I told you I'd see you guys on Wednesday, and here you are on Tuesday, so you must be very dedicated biochemistry students. That's good. Everybody had a good break? No? Studying biochemistry ruined it for you, or what? Other classes? Oh, well, sorry about that. So, a few reminders. Number one, there is an exam in here on Friday at the usual time. As I said, I will not be here. Dr. Mayer will be giving the exam, and he'll have instructions from me on how to give the exam and so forth. The material for the exam will go through what I covered on Friday. It will stop with the lecture material on Friday. Okay? So nothing after Friday's lecture on the exam. Everything I talk about starting today will appear on the final, and the final will be comprehensive for my stuff, as I said last time. It won't include Dr. McFadden's stuff, but it will include everything that I've talked about. Okay? Is that clear? I will do a review session today, and I got confirmation earlier today that we can keep the room. So we'll have the room at 5 o'clock for review. I will keep the camera running, and I'll make a separate video for the review session, and I will plan to get that posted probably tonight. So I have that up as quickly as possible. And that all said, you guys ready to dive into some more? Think about this. This isn't even on this next exam, so now you can just turn your brains off, right? Ha, ha, ha. Okay. Well, I want to say a little bit about something that I didn't get a chance to talk about, which means, of course, that it won't appear on this exam. But it's a very important topic. And that very important topic is respiratory control. Okay? So I've told you that, metabolism starts making sense when you can start putting these individual pieces together. All right? And so that's what I want to spend some time today doing, is putting some of the pieces together relating to metabolism. All right? Now, the mitochondrion is really the center, for our purposes, for metabolism. We'll talk later today about fatty acid oxidation, and fatty acid oxidation will occur in the mitochondrion as well. We'll see fatty acid synthesis occurs in the cytoplasm, so that'll affect things. But fatty acid oxidation occurs in the mitochondrion. So all these energy-producing processes, citric acid cycle, and the fatty acid oxidation, which are two of the biggest energy producers in the cell, are occurring in the mitochondrion. So the mitochondrion is very important. The mitochondrion is interesting in having that inner mitochondrial membrane where those proteins are embedded. So these are the complex 1, complex 2, complex 3, complex 4, complex 5, coenzyme Q, and cytochrome C. All these guys are in the mitochondrial inner membrane. And you've seen the consequences of disrupting the mitochondrial inner membrane. The consequence being that protons can leak back in if you make a hole in it, and if they leak back in, the cell goes crazy. All right? That is, it starts burning all of its energy trying to make ATP and trying to make ATP, but it can't make ATP. And that was the miracle diet drug that we talked about, the 2,4-dinitrophenol, 2,4-DNP. Well, normally our cells don't do that. That is, our cells don't have this out-of-control expenditure of energy. Normally, our mitochondria are what we call tightly coupled. Tightly coupled, okay? 2,4-DNP uncouples mitochondria. And when we say uncouples, what we mean is that the pumping of protons out of the mitochondrion is linked to the synthesis of ATP. That's what, I'm sorry, when it's uncoupled, that's what coupling means. When coupling means that the pumping out of protons is linked to the synthesis of ATP. So uncoupling unlinks those two. It unlinks the pumping of protons and the making of ATP. So 2,4-dinitrophenol, 2,4-DNP, is called an uncoupler because it literally takes these two apart. Okay? Normally, mitochondria are tightly coupled. And what coupling really means is that all of these processes I've talked about are dependent upon each other. They're dependent upon each other. Well, you've sort of seen that already. Because you saw that if the mitochondrion ran out of oxygen, what happened to the concentration of NADH? It increased, right? When there wasn't enough oxygen, NADH couldn't be recycled back to NAD, and so consequently we saw that the glycolysis had to find some tricks to keep going. Citric acid cycle itself couldn't keep going, and that was important. Now, I'd like to think about this in another way, that coupling, that is tightly coupled mitochondria, work, and they work for you. Okay? Let's imagine, if you will, that instead of adding an uncoupler, okay, you're sitting around home, eating pizza, drinking beer, watching the tube. Okay? Eating pizza, drinking beer, watching the tube. Right? Very common American pastime, right? You're not burning any energy. What is your concentration of ATP at that point? It's high, because you're not burning it, right? And you're pouring all this stuff in here that's going to do nothing but make ATP every chance it can. All right? Now, in order for you to understand tight coupling properly, you need to understand two other things that I haven't told you. One is this. Okay? Complex 5 requires ADP. Complex 5 requires ADP. If there is no ADP, that is, if you have high ATP concentrations, then what's going to happen is the protons can't come back in. Protons will be blocked if there's no ADP. Everybody got that? Complex 5 requires ADP. If there's no ADP, then protons can't come in. Okay? The second principle to keep in mind is that the proton gradient, I like to draw the analogy of pumping water uphill. All right? So when I'm pumping protons, I'm pumping water up to, let's say, the second floor. Pump, 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 right? And the more I pump water up to the second floor, the harder it's going to be to continue to pump water up to the second floor. The more the second floor fills with water, it's going to be harder each time. It's eventually going to get to a point where I can't pump anymore. So the gradient of protons will become limiting at some point. The gradient of protons will become limiting at some point, which means I won't be able to pump any more protons. If I can't pump protons, I can't move electrons. These things are all interdependent upon each other. They're all interdependent. All right, so what's happening when I'm sitting around eating pizza, drinking beer, watching the tube? My ATP levels are high. What happens to my ADP levels? They're low, right? What happens to my oxygen consumption? Why is it down? Okay, so it's down because, all right, well, I'm actually jumping a step ahead. My ADP concentrations are low, all right? That means complex 5 is stopped, protons are not coming in. What's happening to the proton gradient? It's getting higher. Proton gradient gets higher, what happens to the movement of electrons? Slows down. When electron movement slows down, what happens to oxygen consumption? It slows down. So when I'm sitting there drinking pizza, eating beer, drinking pizza, drinking beer, watching the tube, what's happening is my oxygen consumption is low. And you know that's the case because when you're sitting there on the tube, you're not breathing nearly as heavily as when you're out there jogging, right? Okay, makes sense? When I'm out there jogging, I change all these. Okay? When I'm out there jogging, what happens to my ATP concentration? It starts going down. I start burning ATP. What happens to my ADP concentration? ADP concentration rises. As ADP concentration rises, what happens to complex 5? Starts spinning. As it starts spinning, what happens to the proton gradient? Proton gradient falls. As proton gradient falls, what happens to the concentration of NADH? It falls because that's the source of electrons, right? Electrons are moving. Therefore, NADH concentration falls, NAD concentration rises. What happens to the citric acid cycle? Starts burning. When I exercise, I burn energy. When I'm not exercising, what's happening to the citric acid cycle? It's stopping. I'm not burning energy. And if I'm not burning energy, what happens to the concentration of acetyl-CoA? I said that last time. You probably don't remember it. That concentration of acetyl-CoA goes up. And as we will see, acetyl-CoA is what we use to make fatty acids. Bang, 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 bang. All of these things are linked. Very, very linked. So I want you, on the final exam, to be able, if I give you a scenario like this, for you to be able to tell me what any of these things are going on. Okay? And if you learn this linkage, it's very simple. You can go step by step by step through there and understand this. Let's put 2,4-DNP in there. What happens? Well, first thing, what happens? What happens to the proton gradient? Proton gradient falls, right? When proton gradient falls, what happens to the synthesis of ATP? Goes down. No gradient to spin the turbine, right? With me? No gradient to spin the turbine. ATP concentrations drop precipitously. Cells using ATP, it's not making ATP. What happens to electron transport? The thing that's stopping electron transport is the gradient. If there's no gradient, electron transport, pushing electrons through. Okay? If electron gradient is going like crazy, what's happening to concentration of NAD? It's going up, right? NADH is the source of electrons. That means NAD concentration is going to go high because NADH is dumping electrons. When NAD concentration goes high, what happens to the citric acid cycle? Spins. This was the idea of the magic diet drug, right? Okay? All right, let's do one more. Then I'll let you guys think about it, okay? Let's imagine that I take some cyanide. I know you guys might like to think about that. Let's imagine I take some cyanide. I take some cyanide. What happens to, what's the first thing that happens to my proton gradient? It's going to go down, all right? Why is it going to go down? Proton gradient is going to go down because ATP is going to be made, but then there's nothing going to be replenishing the proton gradient, right? Because electron transport has stopped, right? Electron transport stops, no more gradient. So ATP concentration is going to go down. How about oxygen consumption? What happens to oxygen consumption? Don't guess, think about it. What's happening to oxygen consumption? Electron transport has stopped. What happens to oxygen consumption? What's electrons go to? Oxygen. If electrons aren't moving, what's happening to oxygen usage? All right? So oxygen consumption goes down, oxygen concentration goes up. Right? What happens to concentration of NADH? It goes up because no place to put electrons. NADH concentration goes up. What happens to the citric acid cycle? It stops, right? And you die. Make sense? Okay, so these things are all linked. These processes are all intertwined with each other. And if we understand how that intertwining happens, we can understand what's happening in our body. I'll remind you of this after we talk about fatty acid oxidation and fatty acid synthesis, okay? Questions before I move forward? Okay. Think about these things. All right. The last thing I will mention here is an important consideration. Okay. I haven't told you this, but I'll tell you now. NADH does not move across the mitochondrial inner membrane. NADH does not move. There's no transporter that will move it across the mitochondrial inner membrane. So those NADHs that are being made out into in the cytoplasm, can't get in by themselves. We have to use a trick to get them in. We use what's called a shuttle, something that carries the electrons and proton from NADH, but not NADH itself. There are two shuttles we talk about. One's called the glycerol phosphate shuttle. Okay? How does it work? Well, it works as follows. Here's the... This thing has decided not to work anymore. There it is. All right. Here are some NADHs. NADHs can't cross by themselves. So what NADHs do is they get donated to dihydroxyacetone phosphate. Where did you see that at? Glycolysis. Excellent. You donate electrons to dihydroxyacetone phosphate, you make something called glycerol phosphate. The cell has a transporter for glycerol phosphate. Glycerol phosphate gets transported into the cell, and then glycerol phosphate gets converted back to dihydroxyacetone phosphate inside the mitochondrion. The problem is that inside the mitochondrion, FAD is converted to FADH2. So we started with an NADH, and we end up with, on the inside, an FADH2. Now I say we collectively because this doesn't happen to us to a significant extent. It's primarily in insects. Insect muscle is full of this transport protein. And the reason that this transport protein is in insect muscle apparently is because it works very, very, very fast. Very fast. Okay? As you'll see, the transport system that we use is different, and it's more complicated. Bottom line here, glycerol phosphate, glycerol 3-phosphate, or glycerol phosphate as it's called, is moved across, carrying the electrons, and then is transported back out as dihydroxyacetone phosphate. So we have a little cycle going here. Each time the cycle turns, more electrons get into the mitochondrion and get left there. Okay? Remember that FADH2 only generates 2 ATPs, NADH generates 3 ATPs. This cycle is not efficient, but it's fast. Okay? If we look at what happens in our cells, it looks a lot more complicated, but it's not. But I'm going to keep it simple. I shouldn't say it's not, but I'll keep it simple. In our cells, we have something called the malate aspartate shuttle. The malate aspartate shuttle. All right? The malate aspartate shuttle works like you see here, and the idea is the same. The electrons from NADH are getting put onto a molecule that gets moved across the mitochondrial inner membrane. Okay? This figure, I think, is a very dumb figure, by the way. We can see that happening right here. In our cells, electrons from NADH are donated to oxaloacetate in the cytoplasm to generate malate. That's the reverse reaction of the citric acid cycle. So we're reducing oxaloacetate to make malate. Malate can cross the mitochondrial membrane. You see this arrow meaning it can go either way. Okay? But in this case, it's going into the mitochondrial matrix. And on the inside of the mitochondrial matrix, the reverse reaction happens. Malate goes to oxaloacetate. Only look what happens in us. NADH is generated. This cycle is efficient. It's not as fast, but it's efficient. You start with an NADH, and you end up with an NADH. You don't lose any energy in the process. The rest of the cycle is balancing the equation. That's all that's involved. All right, now I'm going to say in words what happens. I don't care that you know these words that I'm going to tell you. So you can listen to these words, okay? Malate goes to oxaloacetate. Oxaloacetate is transaminated to become aspartic acid. Okay? Aspartic acid is transported out of the matrix, and on the outside, aspartic acid is deaminated to remake oxaloacetate. What's the source of the amine for the aspartate? Well, it turns out to be glutamic acid. Glutamic acid donates the amine onto oxaloacetate to make aspartate, in process becoming alpha-ketoglutarate. And alpha-ketoglutarate is transported out of the mitochondrion, where it accepts the amine from aspartate to re-become glutamate. Well, they do what?
Draw an arrow from alpha to the blue right at
To here? Yeah, there should be an arrow there, actually. Yeah. Yeah, question. Is that a futile cycle? It's actually not a futile cycle. It's actually a cycle that it's like the other one, that each time it goes around, it's dumping electrons inside of here. So there's something being accomplished every time the cycle turns. Electrons are being dumped inside the mitochondrial matrix, just like they were in the previous one with the glycerol phosphate. So it's not a futile cycle, no. And there's no energy loss either. That's what would happen in a futile cycle. Okay, so what do I care about this? What I care is that here's the important part right here. Malate, oxaloacetate. Electrons carried by malate, and it becomes oxaloacetate. That's really all that matters for our purposes. Okay? The difference being that in our cells, we're starting with an NADH, and we're ending with an NADH. Insect muscle starts with an NADH and ends with an FADH2. It's not efficient. Okay, that is what I want to say about electron transport and oxidative phosphorylation. We are ready to talk about lipid metabolism. Okay. Lipids are very complex. We've already seen examples of lipids that include cholesterol, that include a variety of things, right? They include fats, they include fatty acids, they include glycerol phospholipids, sphingolipids, blah, 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 blah. We're going to focus our attention, for the most part, on fatty acids and cholesterol. We're not going to say much about the others. We'll say a little bit, but not a lot. Okay? Fatty acids are critical. Now, one of the things I noticed that students frequently confuse are fats and fatty acids. Remember that fats are components of fatty acids, but they're not the same thing. I'm sorry. Fatty acids are components of fats, right? But they're not a fat, right? So if I ask you about a fatty acid, make sure I'm talking about that the answer that you're giving me has to do with a fatty acid and not with a fat. And vice versa. If I ask you about a fat, don't give me something that's relative to a fatty acid. Well, fatty acids come from fats, and I've already mentioned this briefly to you, but I'll remind you that fatty acids get released from fats by action of enzymes known as lipases. Okay? These lipases are part of our digestive system, and they occur both inside of cells and also outside of cells. The bottom line being that they release fatty acids from fats. So here's a fat, here are some lipases, and they release fatty acids. That's basically what this is right here. We can also release fatty acids from our membrane lipids by the action of phospholipases. Phospholipases cleave fatty acids off of glycerol phospholipids. So here's a glycerol phospholipid, a phospholipase cleaving it off, and again, we get fatty acid that we can use. So either way, we can get a fatty acid, whether it's from a fat or whether it's from a glycerol phospholipid. Okay. Well, let's follow that fatty acid. Okay? Oh, before we do that, now let's not do that. Let's look at one additional thing, okay? The breakdown of fats is hormonally controlled. The breakdown of fats is hormonally controlled. Okay? It's actually linked to that epinephrine cycle that we talked about relative to glycolysis and gluconeogenesis and glycogen metabolism. It's linked to that. Okay? There is epinephrine, and there's a process, blah, 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 blah. All right? I don't care that you know this process. Okay? That's not the critical point, the critical reason I bring this up. I bring this up to let you know that if I want to cleave off one fatty acid from a fat, that is the very first one, it has to be hormone stimulated. It has to be hormone stimulated. If I don't have that hormone stimulation, I'm not going to break down fat. The second, the third fatty acid, those come off with regular old lipases. But the very first one has to come off with a hormone-stimulated lipase. Okay? And that's what's happening right here. It's trying to show you that you have this, activating the lipase that cleaves the very first one off. The second one, the third one don't require that lipase, and everybody's home free. All right, so imagine I'm a cell and I have just acquired from the bloodstream a fatty acid. That fatty acid, oh, by the way, fat is stored in our body in specialized cells. Not all cells store fat. We store fat in cells called adipose tissue, and they are specially designed to hold fat. That's important because fat is not soluble in water. If it were, all cells would store fat. Okay? So adipose tissue is specifically set up to store fat, which means that when we release energy from fat, that has to be released out of the adipose tissue into the body. Okay. The fatty acid arrives at the cell. The fatty acid turns out to be kind of hazardous for the cell. Fatty acids are hazardous for cells. Why are fatty acids hazardous for cells? Okay. How do we make detergents? Does anybody know how we make detergents? Lye and fat, right? So what you're doing is you're using a strong base and breaking the bond between glycerol and the fatty acids. You're making sodium salts of fatty acids. Okay? What do you know about detergents relative to protein structure? I didn't teach you protein structure. I would have told you what about protein structure? What I would tell you is that detergents denature proteins. And when you denature proteins, they lose their structure, they lose their function. Imagine that I just freely release a whole bunch of fatty acids into a cell. What's going to happen to that cell? It's going to have its proteins get denatured. Not a good career move, right? So cells protect themselves by basically attaching something to the end of the fatty acid. And the thing that they attach is a molecule called coenzyme A. You've seen that before. CoA is what got attached to the acetyl group to make acetyl-CoA. Now we have something called an acyl-CoA, ACYL. Acyl referring to a long-chain fatty acid. So cells are protecting themselves by attaching that fatty acid to a CoA and keeping them from denaturing their own proteins. Good career move, okay? It takes energy to do that. You can see there's ATP that goes in and AMP comes out. That means it's the equivalent of 2 ATPs because there's 2 triphosphates necessary to make this happen. And so cells spend a fair amount of energy doing this, but it's worth it. This also provides a handle. The CoA is a handle for cells to recognize that there's a fatty acid attached to it. Okay? That handle turns out to be very, very useful. Now, here's something that'll surprise you. The cell's gone to all this trouble to put a CoA onto an acyl group. Things get down to the mitochondrion where fatty acid oxidation is going to happen. The fatty acyl-CoA needs to get across the mitochondrial inner membrane, but guess what? It can't. Okay? It cannot get across the mitochondrial inner membrane. So the cell in the cytoplasm has attached this CoA, and it gets to the mitochondrion and goes, hold on, can't do it. Because there's no transport protein that'll take an acyl-CoA across the mitochondrial inner membrane. Kind of dumb, huh? It is kind of dumb. Instead, what happens is in this intermembrane space, that is the space between the outer membrane and the inner membrane, there's an enzyme that will take the CoA off and replace it with something called carnitine. It replaces it with something called carnitine. So instead of having an acyl-CoA, I have an acyl-carnitine. And that can be transported across the mitochondrial inner membrane. And then, when it gets into the matrix, the reverse happens. so that we now make an acyl-CoA again. So what have we done? We have shuttled an acyl group into the mitochondrial matrix. We've shuttled it, and we've shuttled it on the back of a carnitine. The carnitine carried it in. Okay? The oxidation reactions that I'm getting ready to describe to you all happen when the CoA is attached to the fatty acid, not the carnitine. All the carnitine does is the shuttle part. Bear with me? Okay. Well, let's go through the oxidation. Now, I told you when I talked about the citric acid cycle, I said that if you went from succinate all the way up to oxaloacetate, if you learned those steps of the process, you would know the same steps in the process in the oxidation of fatty acids. I'm going to go through these with you, but they're the exact same type of reactions. Here is an acyl-CoA. Okay? If you label the carbons, that's the alpha, beta, gamma. Okay? I'm sorry. That's off. That should be alpha, beta, gamma. It doesn't, in any event, it doesn't matter. I just noticed that. All right? Or you can call them carbons 1, 2, 3. I like to call them carbons 1, 2, and 3. All right? The important thing is that all of the action of fatty acid oxidation and fatty acid synthesis, all of the action is going to occur between carbons 2 and 3. This process is called beta oxidation because it's happening on the beta carbon, but it's really carbons 2 and 3 if we just not use numbers instead of letters. All right. What happens? Well, there's a single bond. The hydrogens and the electrons get removed, just like they got removed from succinate to make fumarate. That happened between carbons 2 and 3 also. Made a trans molecule. There's a trans molecule. Okay? Then we added water across the double bond to make malate. Here's water adding across the double bond. The hydroxyl goes on to carbon number 3. That made L-malate, and look, there's L fatty acid. Then we oxidize that hydroxyl to a ketone to make oxaloacetate, and there's the oxidation of the hydroxyl to make a beta keto acid, just exactly like it was in the citric acid cycle. And then we do something called thiolytic cleavage, meaning we cut off 2 pieces, 2 carbons. Right? That generates acetyl-CoA that you can see down here, and that acetyl-CoA comes from right here. And we have a fatty acid that has 2 fewer carbons. It's not a complicated process. Dehydrogenation, by the way, students in my eCampus class took an exam on this recently, and I had 2 or 3 of them said, there was a question talking about losing water, okay? About losing water, and they said, but this thing does lose water. It loses water right here because that's a dehydration. And I said, you confuse a dehydrogenation with a dehydration. Dehydrogenation means you lose hydrogen. Dehydration means you lose water. Okay? This is a dehydration, okay? An addition of water, oxidation of a hydroxyl, thiolytic cleavage. We've just oxidized the fatty acid. And then we just continue the cycle over and over until the entire thing is broken down, 2 carbons at a time. What about the names? What about the enzymes? I don't care that you know the names. The names are not important to me. Okay? You should be able to understand the basic chemistry that's going on. A dehydrogenation, a hydration, an oxidation, and a thiolytic cleavage. 4 things are happening there. There are 2 enzymes here that are interesting and important. And there are 2 enzymes here that I don't care about. All right? The first enzyme that's important is the very first enzyme. It's called acyl-CoA dehydrogenase. And I tell you that's important. It's important for a very important reason, because we find implications for this enzyme in human health. Acyl-CoA dehydrogenase comes in 3 different forms. One form that works on long fatty acids, one form that works on medium fatty acids, and one form that works on short fatty acids. Okay? Long, medium, short. They all do the same thing. They all pull off hydrogens between carbons 2 and 3. It's just a question of what size of fatty acid they work on. What's the implication for human health? The implication for human health is the medium form appears to be deficient in many children that have sudden infant death syndrome. They're unable to break down medium chain fatty acids. It's not a cause-effect, but it's frequently found that sudden infant death syndrome infants are deficient in that enzyme. Their bodies haven't started making it. The next 2 enzymes, enoyl-CoA hydratase, nah. L-hydroxy-L-CoA dehydrogenase, nah. Okay? They're just names to memorize. They don't really have any importance for us. But the last enzyme, which actually has the simplest name, you can thank me for that, is important because we'll see it popping up in another cycle. It's called thiolase. Thiolase. Okay? Now, I'm not going to tell you about the cycle right now. Just suffice it to say that you should know the name of that enzyme because you are going to see it again later. Okay? And that's fatty acid oxidation. Fatty acids, as we shall see, tend to have even numbers of carbons. So this works out very well. I break off 2 at a time, 2 at a time, 2 at a time, 2 at a time, and I'm eventually left with 2. Okay? That works really, really well. If I have an odd number of carbons, and there are sometimes that happens, then there are some considerations for what happens with an odd number of carbons. Okay? Here's even number of carbons. All right? Stearic acid has 18 carbons. You might say that's 9 cycles, but it's actually only 8 cycles because the last one breaks a 4 in half. You get 2 for that one. So you have 8 cycles to break down 18 carbons, and you generate 9 molecules of acetyl-CoA. And each of those acetyl-CoAs can go into the citric acid cycle and get oxidized. Okay? A fatty acid like stearic acid has a heck of a lot more energy in it than does glucose. Okay? For glucose, we said we got 38 ATPs per glucose if we followed it all the way through when oxygen was present. If we follow all of the energy present, and by the way, I need to show you where we generate some NADs and FADs, NADHs and FADHs. But suffice it to say, okay, that if we completely oxidize palmitic acid, which has 16 carbons, not 18, palmitic acid will generate 134 ATPs. We get a lot more energy out of fatty acids per carbon than we get out of glucose. Why do we bother with glucose? We bother with glucose because glucose can give us very rapid energy. Glucose is soluble in water. It's soluble in our bloodstream very, very easily. Our liver can release a ton of glucose, and it can get to target tissues in seconds. We can't do that with fatty acids. Fatty acids have to be packaged up in something called lipoprotein complexes, and that takes a while. So we need glucose for rapid energy. We need fat and fatty acids for efficient storage of energy. Okay? So fat and fatty acids are for a longer-term energy. Glucose is for very rapid energy. Before I go forward, I want to show you where the FADHs and FADs are made. I forgot to do that. Okay? In this cycle, okay, the very first reaction generates an FADH2. And the third reaction generates an NADH. And these are exactly like the reactions that are in the citric acid cycle because oxidation of succinate generates an FADH2, oxidation of malate generates an NADH. It's exactly like what happens in the citric acid cycle. So each cycle is generating one FADH2 and one NADH. For something like for something like stearic acid, we're going to have 8 of those cycles. We're going to have 8 FADH2s, 8 NADHs are going to be generated. Okay. All right. Now, I said that's what happens if we have an even number of fatty acids. What happens if we have an odd number of fatty acids? We do sometimes have this happen. Okay? We have it happen either by the occasional fatty acid that has an odd number of carbons or by the oxidation of certain amino acids that generate something called propionyl-CoA. What's propionyl-CoA? Propionyl-CoA is a 3-carbon fatty acid. That's what you're going to get if you have something that had 15 carbons. You break down 15, take off 2, you get 13. You get 11, you get 9, you get 7, you get 5, you get 3, and 3 is propionyl-CoA. So if you have an odd-numbered carbon fatty acid, that's one of the things that you're going to get. Well, the cell can't break that down further. So it's got to manipulate it into something that it can break down. And what it does is interesting and very, very odd. Okay? It's interesting and very, very odd. It starts by adding a carboxyl group to it. Okay? So we start with something that's got 3 carbons. We add a carboxyl to it. We put it onto the center carbon. It would have made more sense if we put it onto the end carbon, but we didn't. We put it on the center carbon, and not only that, we put it on in the wrong orientation, so there's actually another step in here where it has to change the orientation of it. And then after it's changed the orientation of it, it says, oh, I want that on the end, and it puts it on the end. Metabolism isn't pretty. Okay? Metabolism wasn't designed by an engineer. Okay? It's not logical to go and do this. But that is what we do. The result of this is we end up with succinyl-CoA. And where do we burn succinyl-CoA? Well, you can read this as well as I can. We oxidize it in the citric acid cycle. So because of this set of reactions, we can metabolize fatty acids and other things that have odd numbers of carbons that produce propionyl-CoA. There's a coenzyme that's necessary for these reactions to occur, and you've heard of it before. It's known as vitamin B12. B12 is necessary for this set of reactions to occur. Okay? This is one of the reasons why we need vitamin B12. Vitamin B12 is the only molecule in our body that I'm aware of that contains cobalt. The atom cobalt. And because of that, cobalt is an essential micronutrient in our bodies. All right. Hold on here. Hold on just a second. I'm trying to decide what to do here. What do you guys think about moving forward or taking a break? I knew the answer to that before I asked it. You're tired of thinking. All right. Well, then the rule is you have to sing loudly in the song. Okay? It's a very easy song. It's called When Acids Get Oxidized. It's to the tune of When Johnny Comes Marching Home. You like that one? Okay. After the song, we'll have a review session. So here's the song. The fatty acids carried by CoA, are oxidized inside the mitochondria. They get to there, as you have seen, by hitching rights on carnitine. Then it goes away when acids get oxidized. Electrons move through membranes. Yes, it's true, it's true. They jump from complex 1 onto CoQ. The action can be quite intense when building proton gradients, and it's good for you when acids get oxidized. The protons pass through complex V, you see. They do this to make lots of ATP. The mechanism you should know goes through the stages LTO. So there's energy when acids get oxidized. That wasn't so bad. Okay. Let's take maybe a 2-minute break, and then we will start the review session. How's that?